This video is related to the experiment 8. Aim: to perform the isolation of pure culture of microorganisms by multiple streak plate technique and serial dilution method. Techniques. References: Tortora, Requirements, Sterile Nutrient Agar Media, Petri Plates, Inoculum. Loop, Micropipette, Burner, Suspension of microbes, etc. Principle. Pure culture is the culture which contains descendants obtained from single patent cell. The microbial population in environment, air, water, soil, includes many species of bacteria, fungi, algae, which usually exists in mixed cultures. To study these microbes, pure culture is required. Generally, two techniques, that is streak and pour plate methods, are used to isolate pure culture. In streak plate method, a loop full of culture is streaked across an agar medium. The streaking is done in different directions. This thins out bacteria and the individuals are separated from each other. In case of pour plate technique, the mixed culture is diluted in the tubes of agar and cooled to the temperature of 40 degrees Celsius. After the dilution process, the agar culture are poured into Petri plates and kept for incubation, and the separated colonies will be observed. Pour plate method was first developed by Robert Koch. The streak plate procedure is another example of isolation technique, and it was developed by Leffer and Gaffey. Multiple streak plate method. The method is based on the principle that by streaking, a dilution gradient gets established across the surface of the Petri plate as bacterial cells are deposited on the agar surface. Because of this dilution gradient, confluent growth takes place on the part of the medium where few bacteria are deposited. Each colony is the progeny of a single microbial cell, thus representing a clone of pure culture. Such colonies may be picked aseptically and re-streaked into fresh media to ensure purity of a particular strain. Serial dilution method. A microorganism that predominates in a mixed culture can be isolated in pure form by a series of dilutions. The inoculum is subjected to serial dilution in a sterile liquid medium, and a large number of tubes of sterile liquid medium are inoculated with aliquots of each successive dilution. The aim of this dilution is to inoculate a series of tubes with a microbial suspension so dilute that there are some tubes showing growth of only one individual microbe. Example: a culture containing 10 milliliters of liquid medium containing 1000 microorganisms, i.e., 100 microorganisms per milliliter of the liquid medium. 1 milliliter of this medium is taken out and mixed with 9 ml of fresh sterile liquid medium, then resulted medium have 100 microorganisms in 10 milliliters or 10 microorganisms per milliliter. Further, 1 milliliter of this suspension is added to another 9 milliliters of fresh sterile liquid medium. Each ml is now contained a single microorganism. If this tube showed any microbial growth, there is a very high probability that this growth has resulted from the introduction of a single microorganism in the medium and represents the pure culture of that microorganism. Next is procedure. Prepare 9 sterile nutrient agar media for the isolating pure culture. On the other hand, make dilution of suspension of microbes by taking 1 milliliter of inoculum and add it into 9 milliliters of sterile water in first test tube. Take 1 milliliter from first test tube and add into second test tube, containing sterile 9 milliliters of water. Then take 1 milliliter from second test tube and add into third test, containing sterile 9 milliliters of water. So, by following this method, prepare 9 dilution test tube and take 0.1 milliliters from each test tube and add to sterile Petri plates. Then, add sterile nutrient agar media to the dilution sample containing Petri plates. Incubate Petri plate for 48 hours. The observations of this experiment could be: pure culture was isolated by using multiple streak method and serial dilution method. This content of slide, you have to write on left side of the practical file, if you are seen this video to prepare your practical file. I request to you, please subscribe this channel and like and share this video to get more videos and updates. Thank you.